Hello, I'm Sofiia and I'm from Ukraine. I'm the author of the project which is called Features of Morphogenesis of the Eyeball of Tailless Amphibians on the examples of Pelophylax ridibundus, or the marsh frog. The main purpose of it was to establish exactly how eye morphogenesis proceeds in tailless amphibian Pelophylax ridibundus up to the complete formation of all eye structures, and how it changes an eyeball at different stages of development. My work included microscopy method. I examined samples of transverse histological sections of the studied individual and observed changes in eye development at different stages. To be accurate, it's needed to mention that the stages of development are defined by Kennedy L. Gostner for a better understanding of these animals' ontogenesis. Don't worry, no frogs were hurt during my research. The samples that I used, they belonged to my city's local university, and they just let me work with them. So what is my contribution? Well, the eye morphogenesis of Pelophylax ridibundus hasn't been studied yet, and using full-fledged samples of frog hatchlings, larvae, and metamorphs, I could see the eye and examine its development from the very beginning to the full formation of an eye structure. I not only observed the changes, but also I measured two main structures, retina and lens, and also the full size of an eyeball at different stages. The main aim was to compare the collected data and to make a conclusion on how do particular structures influence other structures and how do they grow during their development. And for this, I used the special Zen software and the digital camera Axiocam 208, which was connected to the microscope Zen Primostar 3. And using a computer, I could make measurements using the special ruler tool. And it looked like this. Moving to the results of the research and the conclusion, I would say that a lot of work was done and a lot of hours I spent working with the samples. It was a difficult task to measure everything correctly and to describe the morphogenesis correctly, but all in all, I managed to do everything successfully, and the results of the study were approved by my scientific supervisor, who is the candidate of biological sciences, and he is also the head of zoology and histology department in our local university. So what do we have? The results of the study showed that each stage has its own features regarding the formation of certain structures of the eye or changes in their parameters. The morphogenesis of some is affected by the development of others. For example, the bigger the retina becomes, the smaller the vitreous body is. And the size of structures varies sometimes even cyclically, thereby ensuring correct and complete morphogenesis of the eye. Overall, all the details and exact descriptions of each stage are presented in my paper. To sum up, I would say that my research is relevant nowadays because it contributes to the science, especially bacteriology, and also it broadens our knowledge about the widespread species like the marsh frog, native to Europe and parts of Western Asia. And from this perspective, we can make a parallel with the eye development of Pelophylax ridibundus and the human eye development, and study diseases that appear during this process. In other words, the marsh frog can be the model on which we will test different factors in the early stages in real time and see what consequences such influence has. This is since using live human embryos for such experiments is intolerant. Aniridia and coloboma diseases are the perfect examples of eye diseases that may appear during eye development. I can talk more and more about my project because working on it was really interesting for me, and I've always been into learning new things, finding out how particular mechanisms work, how particular systems form, and especially when this is the field of biology. I hope that this project is only the beginning of my research path, and I hope that my research work will continue. Thanks for watching!